This segment is on carbohydrate metabolism. In the last chapter, we learned that carbohydrates and proteins and lipids can be ingested, and then in the small intestine, when they're broken down into smaller monomers, they can be absorbed into the body. And then these smaller substances, these monomers, are the amino acids, lipids like fatty acids and monoglycerides, as well as simple sugars like monosaccharides such as glucose. These organic molecules then will be transported into a cell and will combine in the cell's nutrient pool. Now, once these amino acids and lipids and monosaccharides are in the nutrient pool, one of two things can happen with them. They can be used to build larger molecules, which is called anabolism, and the larger molecules then could be used to maintain and repair the cell, to grow the cell, to allow the cell to secrete substances, and to store those nutrients as nutrient reserves. For instance, glucose can be stored as glycogen. The other thing that can occur with the nutrient pool is called catabolism. Catabolism is when we take these nutrients like amino acids, lipids, and simple sugars and break them down into even smaller substances. So these substances then, if we take glucose and were to break glucose down into smaller and smaller molecules, what would happen is we would be able to generate energy that would provide us with ATP. So glucose, through the process of anaerobic and aerobic metabolism, would yield ATP and heat. As a matter of fact, 40% of the energy gained would be ATP, 60% of the energy would be lost as heat. The ATP is needed in the cell for several different things. The cells they need ATP for locomotion, for contraction, for intracellular transport, for cytokinesis, endocytosis, and exocytosis. These are a lot of the physiological mechanisms that we've seen all semester. Okay, when we look at carbohydrate metabolism, we have to look at the cell. So here's our cell. This is the cytoplasm of the cell. All right, so this is the extracellular fluid out here, or the interstitial fluid. And we have glucose out here. All right. So glucose is going to enter into the cell. So it's in the cytoplasm of the cell. When glucose goes through the process of catabolism, which is the process of breaking it down in order to create ATP, it's going to be converted through a 10-step process into 2 pyruvates, or pyruvate acid. So we get 2 pyruvates. This process is called glycolysis. And it's a 10-step process, and we're not going to need to know all of the steps. There are a couple of things that we need to know about it, though. However, we won't have to know everything for this class. This process, this 10-step process, requires some ATP. So during this process, we use 2 ATP molecules. At the same time, however, during this same process, we are going to produce 4 ATP. So what we can say is, in the process of glycolysis, we have a net of 2 ATP that are created. So we end up with 2 ATP from this process of glycolysis. Now, at the same time, we are going to be able to harvest some hydrogen molecules. So within this compound, this 6-carbon compound, 1, 2, 3, 4, 5, 6. Okay. There are some hydrogen, and those hydrogen can be harvested. And so they will be. By harvesting, I mean that another molecule called NAD will come in like a shuttle bus and pick up hydrogen. And when they pick up hydrogen, the NAD then become NADH. They're actually NADH plus H, but we usually just refer to them as NAD. Now these hydrogen then are going to get transported to a different area in the cell, and that other area is in the mitochondria, and it enters into a process called the Electron Transport System, or Electron Transport System. Okay? And then there it will be used to make more ATP. So we'll talk about the electron transport system in just a little bit, but what we're really interested in are these two pyruvate molecules. If oxygen is present, so if we have O2, if we have oxygen, pyruvate can enter into the mitochondria. And if you remember what the mitochondria looks like, it's one of the organelles within the cell, and we know that it is a double membrane organelle. This organelle is special. It has two layers of membrane, so two phospholipid bilayers. There's the outer membrane, and then there's the inner membrane. The inner membrane is arranged in these folds, which we call cristae. And the fluid inside the inner membrane is called the matrix. It's here in the matrix that we're going to see two different metabolic processes. One is called the Krebs Cycle, otherwise known as the citric acid cycle, otherwise known as the tricarboxylic acid cycle. So we have this one cycle here, and this is going to occur in the matrix of the cell, of the organelle. And then we also have the electron transport system, and this system occurs at cytochromes, which are found embedded in the inner membrane of the cell. Okay, so let's take a look at the Krebs cycle first. Don't forget, we have these two pyruvate molecules that are going to enter into the matrix of the mitochondria and then enter into the Krebs cycle. So let's take a look at the mitochondria. Here's our outer membrane, here's our inner membrane. And pyruvate entered into the cell because oxygen was present. And the first thing that happens is that pyruvate, which is a 3-carbon compound, remember we started with glucose, which was a 6-carbon compound, and it was split in two. And what happens with pyruvate is that one carbon will be cleaved off. So one carbon will kind of be taken out of that molecule, and that carbon will join with oxygen to create carbon dioxide, which is a metabolic byproduct or a waste product. So carbon dioxide is a waste product. This is the carbon dioxide that will be breathed out during respiration. This carbon dioxide, like I said, will exit the cell, it'll diffuse out, enter into the bloodstream, travel back to the lungs, and then you will breathe it out. Now when pyruvic acid loses that carbon, it then becomes acetyl coenzyme A. And this is a 2-carbon compound. Now this 2-carbon compound will be able to join with another 4-carbon compound that's present in the matrix of the mitochondria. And that 4-carbon compound has a name. It's called oxaloacetic acid. And this 4-carbon compound will join with the 2-carbon compound acetyl coenzyme A and become citric acid. Thus, the name of the Krebs cycle is also the citric acid cycle. So now this citric acid is a 6-carbon compound. What we're going to see is that this 6-carbon compound is going to go through several steps. And along the way, 2 carbons will be cleaved from the citric acid. So we'll lose 2 different carbons. And those carbons will combine with oxygen to form carbon dioxide. This is the carbon dioxide, again, that will leave the cell by diffusion, make its way back to the lungs, and then we'll breathe that out during respiration. Okay, so we know that that happens. We lose 2 carbons, so now our citric acid, which was 6 carbons, then becomes a 4-carbon compound again, and as it changes, it actually ends up becoming that same oxaloacetic acid that we started out with. So now the oxaloacetic acid can join with another acetyl-CoA and go through the process again. Since we started out with 2 pyruvates, for every glucose molecule that's split, there will actually be 2 acetyl coenzymes A going through this Krebs cycle. Okay, well, is that the important thing about this Krebs cycle, though, is that we've managed to cleave off 2 carbons and create 2 carbon dioxide molecules? Well, no, not really. We do get a little bit of ATP from the Krebs cycle. So we are able to phosphorylate a molecule that will eventually yield 1 ATP molecule. Because we have 2 acetyl-CoA from 1 glucose molecule, that means that 1 glucose molecule would actually give us 2 ATP from the Krebs cycle. Okay, but still, that's not a lot of ATP. We do have 2 ATP from here, and if you remember back with glycolysis, we also had 2 ATP, but from 1 glucose molecule should give us 36 ATP. And so we have 2 from glycolysis, we have 2 from the Krebs cycle, that only adds up to 4. So where does the other 32 ATP come from? The other thing that happens in this Krebs cycle is that we harvest some more hydrogen. So we are able to cleave off hydrogen in several different places. And 2 hydrogen are cleaved off at each time. And what we find is that in 3 different locations, we are able to harvest 2 hydrogen and have our carrier NAD carry them over to the electron transport chain. So we have 3 NADH molecules that will carry those hydrogen over to the electron transport system. We also have another carrier, and that other carrier is called FAD, and FAD will also be able to carry 2 hydrogen in the form of FADH2, and we have 1 FAD carrier along with the 3 NAD carriers that pick up hydrogen and bring those hydrogen over to the electron transport system. Alright, well, we're getting kind of messy, so I'm gonna just erase this whole board, and let's again take a look inside the mitochondria, and let's just say here's our Krebs cycle. So we have this Krebs cycle here, the TCA cycle or the Krebs cycle, and let's just say that all we're showing here is we have NADH, 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 and we have FADH2. And we have 2 hydrogen molecules from each one of these being transported over to the electron transport system. So each one of these little carriers is going to take those hydrogens over to an area on the inner mitochondrial membrane. And embedded into the inner mitochondrial membrane are these proteins that are called cytochromes. And so NADH will drop off the hydrogen, and the hydrogen ion is going to then split. If we remember the hydrogen is made up of 1 proton in the middle and 1 electron in the outer electron shell. And so if that splits, we end up with a hydrogen proton, and we also end up with an electron. The electron will be released, and the electron will be carried from one cytochrome to the next until it gets to the second to last cytochrome, and at this point it will be released as energy into the matrix of the mitochondria. The energy that it carries will be used to bind a hydrogen that is floating around in the matrix with another hydrogen and also with an oxygen molecule. So here we have H2O, which we know is water. So the byproduct of the electron transport system is water. And remember what we said was the byproduct of the Krebs cycle? We said it was carbon dioxide. Now we should note that both of these processes here require oxygen. So we say that these processes are aerobic. Alright, well, what happens to the hydrogen, to the proton? The proton will actually be pumped across the inner mitochondrial membrane and into the inner membranous space, which is right out in here. Here's the inner mitochondrial membrane. Here's the outer membrane of the mitochondrion. This is the intramembranous space. This is the matrix. This is the inner membrane. And this is the outer membrane over here. Alright. Like I said, we have these cytochromes that are embedded in the inner membrane of the mitochondria. And NADH will bring over the hydrogen to this system, which is called the electron transport system. We said that the electron would be passed from cytochrome to cytochrome, losing energy. When it gets to the second to the last cytochrome, will be released, and the energy will be used to bind 2 hydrogen molecules to 1 oxygen to give us water, and that's our byproduct of the electron transport system. The hydrogen proton is going to be pumped across that inner membrane into the intramembranous space, and after a while, with all of these NADH and the FADH2 bringing the hydrogen over to this electron transport system in the inner membrane, pretty soon we start to build up a high concentration of hydrogen in this space. And there's a lower concentration of hydrogen in the matrix. Well, we start to build up a concentration gradient. Hydrogen is going to have a tendency to want to diffuse across into the matrix. Well, it's allowed to do that in the last protein that's found in this electron transport system. This last protein happens to be an enzyme that's called Atp Synthase, and it has a channel, and through this channel, hydrogen are able to diffuse down their concentration gradient and back into the matrix. And as they do that, they are going to activate this enzyme, ATP synthase. The energy that's used from this ATP synthase will allow an ADP molecule to gain a phosphate group to become phosphorylated into ATP. Now, without getting into all of the numbers here, we can say that from one glucose molecule, there's enough NADH, there's enough hydrogens from NADH and from FADH2 to yield 32 ATP from the electron transport system. The NADH comes from several different places. We are getting hydrogen from glycolysis. If you remember that, we did get 2 hydrogen from glycolysis. We also were able to get some hydrogen in that process of pyruvate to acetyl-CoA, and we are also able to get a lot of hydrogen from the Krebs cycle. Okay, so let's take a look. If we add everything up, we produced 2 ATP directly from glycolysis. And by directly, I'm going to call it substrative, meaning we didn't use the carriers, we didn't transport hydrogen over to the electron transport system. So this is substrative phosphorylation. We also were able to get 2 ATP directly through that same process in the Krebs cycle. And all of the rest of the ATP that were yielded, the other 32 ATP that we made with one glucose molecule, came from the hydrogen that were transported by NAD and FAD in the form of NADH and FADH2. And these hydrogen, again, we received from glycolysis. We also received it from that intermediate step, which was pyruvic acid or pyruvate to acetyl-CoA. And we also were able to yield a lot of hydrogen from the Krebs cycle. And these hydrogen then were able to give us 32 ATP at the electron transport system. So we received 32 ATP from the electron transport system. Overall, grand number of ATP is 32 plus 2 plus 2, which gives us 36 ATP. And that's where we get the 36 ATP from cellular metabolism. Okay, if we take a look at this picture here, so this is showing pyruvic acid, where we're starting from pyruvic acid. Pyruvic acid has one carbon cleaved off of it to form carbon dioxide, and we also see that we are harvesting some hydrogen that will be carried by NAD. This will be carried to the electron transport system. Acetyl-CoA will enter into the Krebs cycle. It will combine with a 4-carbon compound. Acetyl-CoA is a 2-carbon compound, so we will end up with a 6-carbon compound called citric acid. Citric acid will lose 2 carbons and will go through several steps to eventually become the original 4-carbon compound, oxaloacetic acid, that it started out as. It can then join with another acetyl coenzyme A to become citric acid again. Now we know from one glucose molecule we ended up with 2 pyruvates. 2 pyruvates gave us 2 acetyl-CoAs. So we are going to actually use that acetyl-CoA the first time to give us 2 carbon dioxides, and then the other acetyl-CoA will enter into this Krebs cycle to yield 2 more carbon dioxide. So we're yielding quite a bit of carbon dioxide as a byproduct. At the same time, we see that NADH or NAD will pick up 2 hydrogen in 3 different locations, and FAD will pick up 2 hydrogens in 1 location, and all of those hydrogens will be shipped off or carried over to the electron transport system. These carriers will drop off the hydrogen and then go back to the Krebs cycle to pick up more hydrogen to carry it back again to the electron transport system. So it acts kind of like a little shuttle. So we're harvesting a lot of hydrogens from the citric acid cycle. And again, the electron transport system will use these hydrogen to give us 32 ATP. We also see that from one acetyl-CoA we get 1 ATP produced. Because we have 2 acetyl-CoA, we will end up with 2 ATP from the Krebs cycle. And we also had 2 ATP from glycolysis. So the total here is 2 ATP from glycolysis, 2 ATP from the Krebs cycle, 32 ATP from the electron transport system, to give us a complete total of 36 ATP that is yielded from both glycolysis and aerobic respiration in the cell. Here's the electron transport system, and we can see that NADH is bringing over a hydrogen, dropping the hydrogen off, and the hydrogen are going to get pumped across the inner membrane into the intermembrane space, or the intermembranous space. The electron from that hydrogen will be passed from cytochrome to cytochrome and will be released then. The energy from that electron will take 2 hydrogen and combine it with 1 oxygen to form water. And that's why in the electron transport system, water is a byproduct. As the hydrogen, though, are being pumped across that inner membrane into the inner membrane space, it's starting to build up a large quantity of hydrogen in that space. And so we start to develop a concentration gradient where there are more hydrogen in the inner membrane space than there are in the matrix. And so with this concentration gradient, hydrogen is going to have a tendency to want to diffuse back into the matrix. And that's what happens. In that last protein embedded in the inner membrane space of this electron transport system, hydrogen will diffuse across down its concentration gradient. And as it diffuses across, it's going to activate this enzyme. And this enzyme's name is Atp Synthase. As it activates that enzyme is going to give the energy that's required to take ADP and phosphorylate it, or add a phosphate group to it, to create ATP. And in the process of taking one glucose molecule and all of the hydrogen that can be harvested from that glucose molecule and brought over to the electron transport system, we will yield 32 ATP from the electron transport system. This is the basics of what you need to know for this class for carbohydrate metabolism.